Friends, welcome back to my channel, and today in this video we are going to study about hematopoietic stem cell. Before we understand about stem cell and the process of hematopoiesis, let's first see the types of lymphoid organs present in our body. So we have two types of lymphoid organs, one which is known as primary lymphoid organs and second which is known as secondary lymphoid organs. The primary lymphoid organs include bone marrow and the thymus that regulate the development of immune cells from immature precursors, whereas the secondary lymphoid organs, which includes spleen, lymph nodes and specialized sites, they coordinate the encounter of the antigen with antigen-specific lymphocytes and their maturation. That means in the primary lymphoid organs, the cells of our immune system develop and these cells get mature after interaction with an antigen. And this interaction with the antigen happens in the secondary lymphoid organs. So the primary lymphoid organs are responsible for the development of these immune cells, and the secondary lymphoid organs are responsible for these immune cells to encounter the antigens and hence for their maturation. Hematopoiesis is the process by which stem cells are regenerated and from stem cell various other cells are formed. So the stem cell is known as multipotent hematopoietic stem cell. Why multipotent? Because from these stem cells, various other cell lines are formed. So specifically, there are two types of cell lines that formed from these multipotent stem cells. These are myeloid progenitor and the second is lymphoid progenitor. Now from lymphoid progenitors, the cells that are produced are natural killer cells, which is also known as NK cells and small lymphocytes. From these small lymphocytes, the cells produced are T lymphocytes, which are also known as T cells, and B lymphocytes, which are also known as B cells. From B cells, the plasma cells are formed. Plasma cells means these are the cells, that is B cells, that secrete antibodies. From myeloid progenitor, if we see, we get megakaryocyte, from which we get thrombocytes. We also get erythrocyte, mast cells, and myeloblast. From myeloblast, we get basophils, neutrophils, eosinophils, monocytes, and from monocytes we get macrophages. Dendritic cell is the cell which is formed from both myeloid progenitor also and lymphoid progenitor also. So dendritic cells are the cells that are formed from both the lineage, from myeloid side also and from the lymphoid sides also. Now among all these cells in the lymphoid progenitor, the T cells and the B cells, these two cells are the cells of adaptive immunity. Rest all other cells comes under innate immunity. So the T and the B cells that comes under adaptive immunity, rest all the cells comes under innate immunity. And among all the cells, the dendritic cells are the cells that are formed from lymphoid as well as from myeloid progenitor. Now coming to the cells of the immune system, when we are talking about stem cells, so stem cells are defined by two capacities. First, the ability to regenerate, which is known as self-renewal capacity, and the ability to differentiate into all diverse cell types. In the last slide only, we saw this, that stem cell can regenerate themselves, and from stem cell, because these are multipotent, various other cell types are formed. Now, stem cells are of two types, one which is known as embryonic stem cell and the other is known as adult stem cell. Embryonic stem cells are pluripotent in nature. Pluripotent means they can give rise to every cells and organs of the body. So they are pluripotent in nature. Whereas adult stem cells are multipotent in nature. Multipotent means they can only give rise to the cells and the organs in which they are residing. For example, the adult stem cell of brain can only give rise to the cells of the brain. Similarly, the adult stem cell can only give rise to the cells and the organs where they are residing, because they are multipotent, not pluripotent. Pluripotent are the embryonic stem cell, therefore they can give rise to every cells and organs of the body. Now, when no pathogen attacks our body, in that condition, these hematopoietic stem cells, they remain in the quiescent stage. So only small number of these cells they divide. But as soon as the pathogen attack or after chemotherapy, these hematopoietic stem cells, they suddenly shift to the proliferative stage and they start dividing. So the division is very enormous and they undergo a proliferative division, because now they have to kill that pathogen or they have to recover themselves after chemotherapy. So without pathogen attack, or when our body is in a standard resting condition, resting means here the resting is not about exercise, it is about when no pathogen attack. That time the hematopoietic stem cell will divide in a small number. But as soon as the pathogen attacks, they undergo proliferative division. Now let's see the classic experiment to study stem cell. A plate coated with antibodies is used to separate the stem cells and progenitor cells from the rest of the cells. As you can see here, S for stem cells, E for eosinophil, L for lymphocytes, P for progenitor cells. Now each of these cells has some specific markers on their surface. Some markers are common, some markers are different. Now from stem cell we get progenitor cells, from progenitor cells we get differentiated cells like lymphocytes, eosinophils, etc. Now because these are differentiated cells, all these cells possess a marker which is called Lin+. Now this antibody is specific to this particular marker. Now what will happen? As soon as this group of cells are added in this plate and cultured for 1 to 2 hours, because this antibody is specific for Lin+, so all the cells that contain this marker will attach to this antibody. So the remaining cells are the stem cells and the progenitor cells, because these are not differentiated cells, therefore they don't contain this differentiated cell marker. So these cells are then separated, stained and observed under microscope. So this is the process how the stem cell used to be studied. Now, what is the current approach for enrichment of pluripotent stem cell from bone marrow? Now, what it is done in this experiment is, this mouse is lethally irradiated, and when all the cells were added in the body, it was found that the mouse restored hematopoiesis capacity and survived. Now from this, again the antibodies are used to separate the stem cells and progenitor cells from rest of the cells that are differentiated cell. So here what it is undergoing, the negative selection. Negative selection means all the cells that express this marker Lin+ were separated out. So that means this type of selection is called negative selection. Why negative selection? Because suppose we have like this two groups of cells. We want to separate this hollow cells from these stained cells. So when we want to separate these hollow cells from the stem cell, so there are two ways of separating. Either we select this one or we discard this one. These are the two ways for selecting these hollow cells. Now, when we select particularly these cells, it is called positive selection. When we are selecting this cell by discarding the other cells, this type of selection is called negative selection. So here we want stem cell and precursor cells. But to get the stem cell, how we are selecting stem cell? Not by picking up the stem cell, but by discarding other cells. So such selection is known as negative selection. So we are separated. Again, when we injected the remaining cells in the mouse, again the mouse restored hematopoiesis and it survived. Now what we are doing? We are specifically separating this stem cell from the progenitor cells using again some markers which is specific to the stem cell. That is SCA1 and c-KIT. These two markers are specific to stem cell. So now we are separating the stem cell with the use of the markers present in the stem cell. So such selection is known as positive selection. Clear? Earlier what we did? Earlier, the other cells are separated out and that's how the stem cell is selected. So this is a negative selection. Here, the stem cell is specifically selected using the markers present in the stem cell only. So such selection is known as positive selection. So these are the two types of selection. Now what we can see in this graph is, this is the increasing number of cells. So here is the survival rate given. Now, when we are using all the cells, including stem cell, precursors and differentiated cells to the mouse, so that it can restore hematopoiesis, we observe that large number of cells are needed so that the mouse can restore hematopoiesis. Then when we separated the differentiated cells, now we have the second one is for the stem cell and the precursor cell. We have only two types of cell now with us, stem cells and the precursor cell. So we required less number of cells to restore hematopoiesis. And when we separated out only stem cell, then we saw that very less amount of cells is also sufficient to restore hematopoiesis. Clear? So that means for hematopoiesis, what is needed? The stem cell is needed. So if we give the purified stem cell to anybody, so very less amount of stem cell can also able to restore the process of hematopoiesis. But if we cannot purify and if we give the whole cell, whole bone marrow, then it will take lot of cells to restore the hematopoiesis. So that's how we can say that how much the stem cell is important and it is pluripotent in nature for the regeneration of stem cell as well as the other cell lineage. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.